In order to use your miniPCR Bio blueGel electrophoresis system, you'll need to pour agarose gels. miniPCR Bio provides 3 options for pouring gels: all-in-one agarose tabs, plain agarose tabs, or agarose powder. This video will cover how to create gels using agarose powder. You can find links to videos on the other 2 methods in the description below. First, you'll want to ensure that you have an appropriate quantity of TBE buffer to run and pour your gels. You will need 20 milliliters of buffer to make each gel and 30 milliliters of buffer to run each gel. TBE powder from miniPCR Bio makes buffer preparation simple. For most classrooms, the 5.1 gram container of TBE powder, which makes 600 milliliters of TBE buffer, enough for 10 blueGel runs, is sufficient. Combine the TBE buffer powder with distilled water and mix until dissolved. It can take up to 15 minutes for the powder to dissolve, but you can stir or gently heat the solution to help the powder dissolve more quickly. Before pouring your gel, you want to set up your gel casting platform. Place the clear gel tray into the white casting platform. Place the comb in the notches of the platform at the end of the gel tray. For users new to pipetting, we recommend placing the larger side of the comb with fewer teeth down. If you need more wells in your gel, you can add a second comb to the second set of notches. Now that the casting platform is set up, we can make our gel. In this example, we will be making a 2% agarose gel. Using agarose powder is the traditional way to pour gels. For a 2% gel, we will use 0.2 grams of agarose for every 10 milliliters of buffer. Because we want to make a single 20 milliliter gel, we will measure out 0.4 grams of agarose powder. You can find detailed instructions on the amount of agarose needed to pour gels of other percentages in the description below. Combine the agarose powder with 20 milliliters of TBE buffer in a container with at least 100 milliliters capacity. Heat in a microwave for 60 seconds or until the solution boils. Keep an eye on it to make sure it doesn't boil over. Swirl using a heat-proof mitt to make sure that the solution is well mixed. Make sure that all of the agarose crystals have dissolved. If agarose crystals remain, microwave for another 5 to 10 seconds, then check again. You can also use a hot plate if a microwave is not available, but this will take longer. Once your agarose solution is dissolved, add 1 microliter of a fluorescent DNA stain, like SeeGreen or gel green, for every 10 milliliters of gel. Here, because we made a 20 milliliter gel, we will add 2 microliters of a fluorescent DNA stain and swirl to mix. Once you have prepared the agarose solution, pour it into the assembled gel casting tray. You can pour the agarose solution immediately into the tray. You do not need to wait for it to cool. The gel will set in about 10 minutes. Once cooled, it should feel firm like Jell-O. Now you're ready to remove the combs and load your gel into your electrophoresis box. If you want to store your gels to run at a later time, you can prepare gels up to 5 days in advance. Wrap your prepared gels in plastic wrap or place them in an airtight container and store at room temperature, protected from light, for up to 5 days. Gels should not be stored submerged in buffer. To learn more about each method of pouring gels meant for the blueGel electrophoresis system, you can visit minipcr.com.